All right, thank you, Rob, for the kind introduction. So my name is Chun Yu. I'm an associate research scientist. So I will present you the data we recently published on human KIT-positive myeloid cell facilitating visceral metastasis by melanoma. So melanoma metastasis is a spread of cancer from one organ of the body to another not directly associated. And it accounts for most of the cancer-related mortality. However, most of the studies on the tumor metastasis are based on syngeneic or genetic modified mouse model, germ model, or on the human tumor that are implanted in an immunodeficient mice like MISTRG that doesn't have a human immune system. And this type of experimental model often is sufficient to model the interplay between the human tumor and the human immune system, which we think that is crucially important in the context of the cancer development. So, and this is particularly true, we believe that in the melanoma. So melanoma can metastasize to all parts of the human body. And when you look at the survival, the non-pulmonary visceral metastasis, the metastasis go to the spleen and go to the liver, usually have a worse prognosis than the tumor that go to the lung. And the new treatment, including the target therapy with BRAF, MERK inhibitor, or immunotherapy like anti-PD-1 or anti-CTLA-4, this has been dramatically improved the survival. However, with this new treatment, there's still a significant fraction of patients that either do not respond to therapy or they will succumb to a treatment-resistant disease. And this is often due to dissemination of the growth of this tumor. And then this is driven by a very complex interplay between the cancer cell and microenvironment. And this type of mechanism are still poorly defined. So with this, we set a goal to first delineate the micro environment in the tumor and to see what type of immune cell are actually present in human melanoma. So with that, we first use RNA sequencing data. Using the cybersol method, we can estimate the fraction of each immune cell type present in the tumor. And this is a method that without involving a dissociation that often destroy myeloid cell in the tumor. And so with this method, you could see that there's different cell type are present in the tumor bed, and myeloid cell account for 50% of total immune infiltrate in the tumor bed. And furthermore, we want to know what type of immune activation are present in this tumor. So with that, we use immune module analysis. So this analysis allows us to define the function of this immune cell. And so with this analysis, we can cluster the patient sample into hot tumor and cold tumor. And this is basically, you could see it through the heat map that the hot tumor often has B cell transcript, the NK cytotoxic transcript, inflammation, interferon, and T cell transcript. However, when you look careful on the myeloid transcript, there's really no distinct separation between hot and cold tumor. And this is also found in the TCGA sample. So when we look at the TCGA sample, that we also found a majority of immune cell are myeloid. And if you look careful for the CD33, the CD33 is a pan-myeloid marker. So when we look at CD33 transcript, there's indeed more of CD33 transcript in the metastasis than the primary tumor. And this is reflecting in the immunofluorescence staining, where we can find the CD33 positive red cell are present in the tumor bed. So how do we study human immune cell, myeloid cell particular, in vivo? So there's two humanized mouse model that has a feature of myeloid compartment. So one is MISTRG. The mice has genetic expression for human MCSF, GM-CSF, IL-3, and SERV alpha and TPO. So because the presence of MCSF in this model, so they the mice develop CSF1 dependent myeloid compartment. And we have shown together with Richard Favell from the Yale University that this type of mice has accelerated primary melanoma tumor growth that is linking to the presence of human macrophage infiltration. Another model is NSG SGM3. The mice has transgenic expression of stem cell factor, GM-CSF, and IL-3. Because the lack of MCSF, this mice develop CSF1 independent myeloid compartment, and has been shown that they have broadened myeloid and CD4 positive T cell. And indeed, when we transplant these mice with CD34 HPC and look at the myeloid compartment and compare to the NSG mice, so what we see is there's higher fraction of CD33 positive myeloid cell in bone marrow, spleen, and in the blood, and in the peripheral like lung. And when we phenotype this myeloid compartment using multi-color flow cytometry and using reduction using t-SNE, we're able to see that the myeloid cell between NSG and SGM3 are really segregated. And then this is a feature by higher expression of CD11B, 11C, and CD14, and CD66B, and CD117. So phenotypically, the cells that are present in SGM3 are indeed different from the NSG. And transcriptionally, when we look at RNA sequencing for the gene expression, they are also very different from the NSG. So we can perform differential gene expression, and then we can find that different genes are expressed in SGM3 versus NSG. And if we are looking at the function using the IPA analysis, what we found is SGM3 mice, the myeloid cell are more featured in granulocyte or granulocyte adhesion and diapetasis and Th2 pathway, while the NSG has higher expression on the Th1 pathway at IL-15 production. So, how do these two mice respond to human melanoma? As I'm being told you earlier that the MISTRG mice, when we transplant the melanoma tumor, there's a progression of tumor growth. But when we implanted the same tumor cell into the SGM3, we did not see any difference in the primary tumor growth, as you see here. However, when we look at using the tumor growth using in vivo imaging, so we label the tumor cell with luciferase and then follow the tumor progression, what we see is we start seeing a tumor grow in the distant side, start at day 35 after tumor implantation, and so forth. And this is also consistent when we perform the necropsy. At 8 weeks post implantation, we found a significant higher fraction of mice with macroscopic tumor that grow in the spleen and liver. And to describe the extent of this metastasis, so we use ex vivo imaging system. So you could see that from the heat map plot that in addition to the spleen and liver, this melanoma can spread to different parts of the mice. So including lymph node, the spleen and liver, and then pancreas, stomach occasionally, in the kidney, in the lung, and the bone. So we haven't found the tumor grow in the bone yet. It's probably due to the lack of myeloid cell infiltration in this model. That's why. So with this, we ask the question, is this dependent on the transgenic expression of the cytokine, or a human cell are indeed involved in this metastasis? So with that, we basically perform a very simple experiment comparing humanized versus non-humanized mice. And then we follow the tumor progression. So what we found is very as expected, we didn't see a difference in tumor growth. However, the tumor metastasis in the spleen and liver was found in humanized mice, but not SGM3 non-humanized mice. And this was also been confirmed by immunofluorescence staining that the tumor was grown next to the CD45 positive human cell. So because as I mentioned earlier, this mice has the SGM3 cytokine. So we asked whether the cytokine are important in this context. So what we did here is we performed an adoptive cell transfer experiment. So in this case, we purify. So we first generate humanized SGM3 mice, and we purify the total leukocyte from the humanized mice and transfer to a naive non-humanized SGM3 or NSG mice. And then we follow tumor progression in these mice. So as expected, when the mice, the non-humanized SGM3, received the CD45 cell from a humanized mice, and they develop metastasis. However, when we implant this same cell, pro-metastatic cell, into NSG, we did not observe any metastasis in this model, suggesting that the continued presence of these three cytokines may be important, and the nature of this pro-metastatic cell may require this cytokine to exert their function. So as you recall earlier from the immunofluorescence staining of the human tumor, that we see a lot of CD33 myeloid cell also infiltrate in the experimental tumor in the liver. And then when we look at the transcriptome of this CD33 and compare to the transcriptome in the patient tumor, we actually see over than 78% homology, not homology, like, common expression between these two sample types, which confirm that, we are heading actually the right direction, that the CD33 in the SGM3 may be very similar to the patient tumor. And this is driven by a step 1, step 3, and NF-kappa B. So, of course, the consequent question that we want to know is whether these CD33 are indeed, promoting the tumor growth. So to do that, we basically fraction the CD45 cell into CD33 positive total myeloid cell versus CD33 negative lymphoid cell, and transplant them to the non-humanized mice and follow tumor progression. And as we expected, the cell that the mice that receive CD33 positive but not CD33 negative develop metastatic tumor in the spleen and the liver. So as you recall, there's a big fraction of myeloid cell I actually express CD11B. So we first looked into the CD11B compartment. And so we found that using the same adoptive cell transfer experiment, the cell, the pro-metastatic cell, is actually reside in the CD11B positive, not the CD11B negative. And when we look at the different subset under the CD11B compartment, we know that there's CD66B granulocyte or CD14 monocyte, as well as CD117. So because the limit of time, I will not focus on CD66B and 14. And these two cells, we use the same adoptive transfer, has shown that these are dispensable from our model. And so we focus rather than CD117. So CD117 is often expressed on mature mast cell. So with that, we take advantage of another humanized mouse model, is NSG with a transgenic expression of membrane stem cell factor. Here for sure for HM-CSF, the one in green. So this mouse has shown to have developed tryptase positive mature mast cell when they become humanized. So we implanted the same tumor into this mice. And then as expected, we didn't see any difference in the primary tumor. However, when we look at the liver metastasis, these mice are unable to develop liver metastasis, suggesting that the mature tryptase positive cell may not be important in the context of visceral metastasis. And this is consistent with our finding using the adoptive cell transfer experiment. When we deplete the human FCER1A positive cell, we're still able to identify and found the liver metastasis. So suggesting that the mature human mast cell do not promote visceral metastasis. So with that, we came back to the CD117 and we separate the cell into CD117 positive and negative cell. And indeed, we found that the CD117 are responsible for the visceral organ metastasis, as shown here that the once we deplete CD117, there's no longer has a capacity. So suggesting that this CD33 positive, 11B positive, and CD117 positive cell are indeed responsible for the visceral organ colonization. So to put this our finding in the context of human melanoma, so we want to define a transcriptome that describes this CD117 positive cell. So with that, we actually utilize the single cell RNA sequencing data. So here we basically purify the cell from CD33 cell from this prometastatic environment, and then you perform a single cell sequencing. And of course, we can find different subset of, immune like myeloid cell in this. And so to define the transcriptome of KIT positive cell, so what we did is we segregate KIT positive versus KIT negative. KIT is the gene for transcribed CD117. And so we compare this KIT positive versus KIT negative cell in this single cell RNA sequencing data. We're able to develop, generate a differential expressed gene list. And using this differential expressed gene, we can generate a KIT score that represents a transcriptome of KIT positive cell. And we then apply this KIT score into a TCGA sample. And then, so with this sample, we stratify into KIT score high sample versus KIT score low sample. As you've seen in the survival probability, the KIT score high has a lower survival probability. And this is consistent with our data in the mouse model showing that the KIT positive cell or CD117 positive cell are actually responsible for visceral metastasis. So in summary, what I show you here today is we utilize the humanized mouse model. We found that there's a KIT positive cell under the influence of stem cell factor, GM-CSF and IL-3. This cell actually can promote the melanoma cell growth in the visceral organ. And we believe that this can be a novel candidate for biomarker or a therapeutic opportunity for metastatic melanoma. So with that, I would like to thank all the people contribute to this work. So the work is led by Dr. Carolina Peruca at Jackson Laboratory with the help from different members of the lab. And this work is in collaboration with many members of the Jackson Laboratory. And the funding is provided by Jackson, the Director Initiate Fund, and then the NIH as well as Department of Defense. With that, thank you for your attention.